edition of Intu's Webinar Wednesday. Chelsea's on holiday today, so I'll be your host. My name is Michael Alpaugh, and I'm the Intu US East Asia Recruitment Manager based here in beautiful Ho Chi Minh City, Vietnam. This week, we joined two nationally recognized experts from St. Louis University about the search for a COVID-19 vaccine. Dr. Daniel Hoff is SLU's Director of the Division of Infectious Diseases, Allergy, and Immunology. He also runs SLU Center for Vaccine Development and serves as the Director for the Institute for Vaccine Science and Policy. Dr. Sarah George is a SLU Associate Professor of Infectious Diseases, Allergy, and Immunology. She is the university's principal investigator for a cure for COVID-19. During the video, please feel free to type your questions and comments to me. I'll try to get to as many as I can, and if I can't help you, an Intu representative will reply to you personally during the week. Also, before I begin, if you'd like to enjoy today's program at your own speed and also view many other talks from Webinar Wednesday, please look for the link in the follow-up email from today. All right, let's begin.
With us tonight, Dr. Daniel Hoff is the principal investigator of SLU's Vaccine Treatment Evaluation Unit, also known as the VTEU, and he's director of the Division of Infectious Diseases, Allergy, and Immunology at SLU. He is also director of the Center for Vaccine Development. And Dr. Sarah George is an associate professor of infectious diseases, allergy, and immunology at SLU, and is the principal investigator of a COVID-19 treatment being conducted right now at St. Louis University. First, let me start with you, Dr. Hoff. You're the head of SLU Center for Vaccine Development, which houses one of the country's only 9 elite VTEUs around the country. That means SLU is on the front lines in developing new vaccines and treatments for infectious diseases. What is a VTEU and what role does SLU play in helping develop a vaccine for COVID-19 and other diseases?
What a VTEU stands for Vaccine and Treatment Evaluation Unit. This is a designation that is given to sites that are awarded NIH initially contracts, now in the new cycle grant, which fund work to develop vaccines for pretty much everything except for HIV in human clinical trials, phase 1 through phase 4. So the 9 units that are currently funded work together to try to develop new vaccines to protect the U.S. public and also the world. The other major function that we serve is in national preparedness, which is very relevant right now. We have been involved in multiple efforts to try to protect the U.S. public against imminent threats or ongoing threats from the time of after 9/11 and concerns about bioweapons to the 2009 H1N1 pandemic to avian flu scares, Zika, and now COVID-19. So that is really what the VTEU network does and what we provide in value to the U.S. public.
All right, thank you, Dr. Hoff. Dr. George, you're leading the study of an investigational treatment for COVID-19. Can you tell us about the study and where it stands at this point?
Well, thank you. This is an extremely important study. This is sponsored by the National Institutes of Health, and we are running it here at St. Louis University Hospital. This is a study of a promising antiviral drug called remdesivir made by Gilead, which is being studied in people hospitalized at St. Louis University Hospital with COVID-19 disease. This drug looks very promising in animal models of viruses closely related to COVID, the coronavirus causing the current outbreak. And there has been recently published, including just today, some very promising, though not definitive data, looking like this drug may be very promising. So we're very glad to have this trial. and we're going on with it, and it will continue for weeks to come. Thank you.
Dr. George, if I could follow up, is this the drug that people may have heard about that was used to help treat Ebola?
Yes, this was used in some Ebola treatment trials in the outbreaks in Africa.
There has been a lot written and said that it takes a minimum of 18 months, perhaps longer, to develop and make ready a vaccine for a virus like COVID-19 or other viruses. Can you talk about why it takes that long? I think people wonder why it can't be done quicker than that.
The two main things that you have to do when you're developing a vaccine for use in humans is very carefully study the safety of the vaccine and very carefully study the effects of that vaccine in terms of being able to provide a protective immune response. To do this, you start with, of course, there's a lot of work done before you ever get into humans. You have to identify what molecule from a pathogen could be used, or the whole pathogen, in what form it could be used for a vaccine. You have to do animal studies to see if it works, and there's no major toxicity issues. And then you have to make the vaccine in a way that it can ramp up to large numbers of doses, so it would be useful for the long term once you get through the clinical trials. That's not always easy. Sometimes vaccines fail at the step of trying to scale up. After getting GMP material, good manufacturing practice material, which has been generated in a way to minimize chances to almost zero that there's any contamination that could cause any harm to anyone. This is then the time when you can start thinking about doing clinical trials in humans. You start with a few. A phase 1 trial usually just has maybe a few dozen individuals or up to 40 or 50 subjects. And what you're really looking for in the first in-human trial is whether or not there's any really big safety problems. Of course, you're also looking at whether or not the vaccine induces any immune response. But you're not getting at the heart of the matter of whether or not it can actually protect the population against the disease it's intended to. So you move on to phase 2, assuming there's no big safety signals in phase 1, and you see some signal of immunogenicity or inducing an immune response. And in phase 2, you have larger numbers of people. And the same thing happens going into phase 3. So you progressively go from small numbers of subjects to larger numbers, and then large enough numbers to where you can have the statistical power to look at whether or not this vaccine really does protect people significantly. So all that takes a lot of time to do it right, to do it safe, and to make sure that we know whether or not the vaccine can work.
Great, thank you. Dr. George, a number of people submitted questions around this question about whether a person's blood type has any impact on whether they are more or less likely to get COVID-19 or provide any protection.
Well, thank you. So I think that comes out of some data from China where they looked at blood types of people hospitalized with COVID-19 and then also blood types in the surrounding region. And they showed that people with blood type O were underrepresented in the hospitalized population relative to the surrounding population. However, there certainly were people with blood type O who were hospitalized with COVID disease. And also, they only looked at people who were sick enough to be hospitalized, not those who were well enough to stay home. So the data is intriguing, but it's not definitive. And please do not think that if you have blood type O, you're protected. You're not.
Thank you. I'm going to follow up, Dr. George, with you again. There has been a lot of discussion in the media and in other places about the drug hydroxychloroquine as a possible treatment. Can you give us your perspective on whether there are trials taking place of that drug and where they stand at this point.
Well, the good news is that there are trials taking place of hydroxychloroquine, which has been much hyped and I would say overhyped in the media. And we need to do those trials, just like the study we're doing now at St. Louis University with remdesivir, where we really need to get accurate data to see if something actually works. And that means we have to compare it with just supportive care only. I shouldn't say just supportive care, because supportive care is actually the only thing that's really approved for COVID disease at this time. To date, the studies published on hydroxychloroquine really don't look very promising, including some very recent data that's just come out where it just doesn't look like it's having much, if any, effect at all. So again, right now, there's no real data to show that it's a beneficial drug, and we just really need to do the studies and understand it one way or the other.
Great. Hey, Dan?
Yeah, just if I could add, the other issue that has come up is that there are a lot of patients that use hydroxychloroquine, and we need chloroquine for malaria treatment, and patients with autoimmune diseases tend to take hydroxychloroquine, and chloroquine is good for the malaria infection. So those indications are very important, and there's limited amounts of these drugs. So it is an issue if people start using a lot of a drug, particularly if it's not shown to work. It takes away from the ability to treat other people with other significant diseases, because there are limited amounts of these drugs.
And again, because it's been so talked about, I've also read and heard stories about some side effects from the use of hydroxychloroquine, including possible heart issues. Dr. George?
Yes, that's a very good point. Thank you for making that point. Hydroxychloroquine does have some very real effects on the heart, particularly people with heart disease. And as has been discussed, people with heart disease are among the groups most vulnerable to COVID-19 disease. So that drug has very real toxicities. And again, all the more reason it's important to really do proper studies of it and see whether or not it works.
Great, thank you. Dr. Hoff, there seems to be some discussion and confusion over whether those who have contracted COVID-19 are then immune from contracting the virus again. I've heard it both ways. What can you talk a little bit about that?
Certainly. At this point, we don't really know. We don't have sufficient information, but most people in the field do believe that the infection itself will induce a protective immune response in most people. Otherwise, you wouldn't clear the virus. Other evidence that suggests that this happens commonly is we have already seen in multiple places in the world that there have been major outbreaks of this infection. But then it seems to slow down, and China has completely reduced to very low levels, and now they're more focused on preventing people from coming into their country with the infection and reintroducing it. So there's that type of, I guess, circumstantial evidence that would suggest that people do develop an immune response that is protective. How good that immune response is, we don't know. Most people in their middle ages, when they have a healthy immune response, will develop a strong immune response to an infection after they develop and while they're clearing it. That usually does provide protective immunity. In fact, I don't know of any good examples to the contrary. But what we don't know is in people that don't have strong immune responses, if they get an infection and they're lucky enough to get over it, then will they be as protected as other individuals? So one point is that it's likely that there's a gradation of the level of protective immunity, and that develops after infection. In the bulk of the population that are healthy, The immunity should be good and last at least for several months to a year. And we're hopeful that it might be longer than that, but we don't know at this point. The other issue that's related to this is, how do we measure immunity? We don't really know what's protective. We do think, based upon a lot of animal studies, vaccine studies, that antibodies directed against a specific part of the virus that's required to engage a cell that it infects and then initiates the infection. If there are antibodies that can block and bind specifically to that portion of the virus and block the interaction between the virus and the target cell, then you can protect against that cell getting infected. There's T cell responses, which are a different type of immune response that those cells don't make antibodies, molecules floating around that can block things like viruses from infecting cells. They actually recognize infected cells and can destroy them. So that's another part of the immune response that is important in protective immunity, and probably is important here too. The tests that are coming out that look at antibody responses and trying to screen people are very new, and we don't know exactly how to use them yet. There could be problems with specificity because we've all been infected with common coronaviruses that circulate every year. And some of these antibody tests may just measure some of the antibodies that preceded the entire COVID-19 problem so far. So there's a lot of unknowns. We do think that most people will have a protective immune response after they clear the infection, but some people may not. And for how long that immunity will persist, we don't know either.
Another question, Dr. Hoff, for you. What about the possibility of giving people plasma or antibodies from someone who has been infected with COVID and recovered? Mm-hmm. I'm assuming there are some studies being done of that as well?
Yes, there's very many. There's several that have already been done, and there's a big effort here in the U.S. to try to have volunteers that had gotten COVID-19 and recovered clinically. And if you take serum from those individuals, from their blood, and infuse that into people during an acute infection with SARS-CoV-2, theoretically, they can be protective. This has been it's called passive immunotherapy, which just means instead of actively immunizing or infecting to try to get an immune response, you're transferring these soluble antibody molecules into somebody so they have a preformed immune response. In fact, there's a large multi-center study that our hospital system is planning to be part of, that's being led by the Mayo Clinic after they received emergency use authorization for doing this type of passive immunotherapy. What we don't know about it, though, is what does the level of antibodies that you're transferring in need to be to provide optimal protection. So one thing important to be looking at is, in doing studies like this, always looking at what the antibody titer is that's present, and then learning from the experience of the clinical outcomes what the level needs to be to provide satisfactory protection. Another way of doing this in the modern age is to make what's called monoclonal antibodies. These are antibodies, just a single molecule that's made from one cell, B cell, that is the type of cell that makes antibodies. Lots of companies are making these now. There are cocktails coming out for study. And we've used a lot of monoclonal antibodies in treating people. These are also termed biologics, or things that we use to block inflammatory responses in autoimmune disease, like blocking TNF in patients with autoimmune disease. Antibodies directed against the virus specifically that can block it, like I described before, from infecting a cell, could be very useful and could more easily be reproducible in terms of the optimal dose that could provide the best protection. So these are things that we are planning to study here as we go forward in this pandemic.
Maybe we'll finish on a higher a positive note. I'd like to ask both of you, what gives you optimism for the future in dealing with this with COVID-19? Dr. George, I'll start with you this time.
Well, we've beaten some very tough viruses and pathogens before. You know, we now have a licensed vaccine for Ebola. We did not have that with the African Ebola outbreak, which I guess was the last big world public health scare. We do have a lot of testing coming online now. We do, as Dr. Hoff said, have antibody testing now just starting to be developed and become available. We've beaten these things before, but it's going to take time, and it's going to take good data and good studies.
Dr. Hoff, what makes you optimistic?
Well, I would say the same things as Sarah, but I would also say that it's just so wonderful to see how many people care about everyone else and are doing everything they can when we don't have a vaccine, we don't have a drug yet. Following social distancing is incredibly important, and everyone is a hero that's doing that. And that fills me with optimism that our American public, the world citizenry, are coming together and trying to help each other and make a big impact on this. We will develop vaccines and drugs that are effective. Can't say exactly when we will have them, but in the meantime, if we work together, we can all fight and beat this thing.
That's a great way to conclude. Thank you. Dr. Sarah George and Dr. Dan Hoff, we all appreciate and thank you for the work that you and your teams are doing to try to find answers to this virus. And we thank you both for taking the time to be with us. These are two of the renowned experts in the country in dealing with these types of diseases. So thank you again for being with us. And
Right, great interview there, and some positive news at the end. I think I speak for everyone when I say that I hope the team at SLU is able to help find a cure for COVID-19 soon. All right, everyone. I hope you enjoyed today's discussion, and that's all I have for you. Please remember to share the registration link with your colleagues and students, and be sure to tune in next Wednesday where we'll have a look at engineering at OSU. Thank you very much, and goodbye.